So welcome back to part 2. I now want to talk a little about what a cell is made of and the fundamental structural components of cells. But before I do, just a quick reminder of what we did in our last lecture. We talked about cells, we understood how they are relative to size of tissues and organs. We talked about cells being building blocks. They are the fundamental building stone of bodies and humans. We talked about how they come in all sorts of shapes, squamous, cuboidal, columnar. We talked about how they come in different arrangements, and we reminded ourselves about the issues of looking at scale and the worry of looking at sections of three-dimensional objects just in 2 dimensions. Please, if you get a minute, do watch some of the work on the Khan Academy. There is some fantastic work around understanding how cells are. There's plenty of good material around to look at. Now, the first part of a cell I'd like to talk about is the cell membrane. This is the bag that contains the intracellular components and keeps the outside environment out. So the cell membrane is the outer limit of a cell. A cell membrane is fundamentally made of fat, but in that fat is embedded a series of proteins, and these proteins have a very critical role in allowing the cell to interact with its outside world. And just remember that this cell membrane separates the outer world from the inner world. And here's a diagrammatic representation of what it looks like. It's actually 2 sheets of phospholipid or fat. The phospholipid on one end, on one surface, is water soluble or hydrophilic, and the other surface is hydrophobic. And because we pack 2 sheets together like a sandwich, the 2 hydrophobic areas are back to back, and the 2 hydrophilic areas face the extracellular environment or the intracellular environment. This is a very clever arrangement because it allows cells to exist in a watery environment, but at the same time, by having the internal fat, it helps to lock the external environment from the internal environment and therefore regulate the interaction between the two. And floating in this bilaminar phospholipid is a series of proteins. And these proteins poke from one side to the other or are purely on one side. And these proteins provide a number of roles in the cell membrane. For example, some of the proteins sticking on the external surface of your cells are like number plates. They uniquely identify you. And therefore, that's what prevents us from taking a liver from anyone and putting it in anyone else. It's these proteins on the surface of your cells that are unique markers to you that defend you against transplants, or more importantly, every day of the year, they defend you against foreign materials such as bacteria. Other proteins in the cell membrane are there to transport things from the external world to the internal world, or vice versa. So there is a whole bunch of proteins responsible for transport that go across the cell membrane. And there are thousands of other proteins. We are still finding today different proteins in the cell membrane and the different roles that they take on. And here we have 2 cells, a blue cell and a green cell, coming up right next to each other. And this has been taken with a transmission electron microscope at very high magnification, so you can actually see, if you look very closely, the 2 layers of each of the cell membranes on each of the cells and the Brown in between is the extracellular environment. So this is a very common arrangement you see in the cellular world. And the surface membrane of a cell can have all sorts of interesting projections from it in two fundamental ways. They can have things called microvilli, and microvilli just increase the surface area of the cell. And if you have more surface area, that means you have more area for those proteins embedded in the cell membrane to absorb or to excrete materials through the cell membrane. So microvilli are fundamentally there to allow you to increase the pace of communications between the internal and external environments of a cell. Whereas on the left, and do notice the little red line in the red circle is to give you a scale. That little red line is the equivalent size of a microvilli. So you can see these things on the left called cilia are many, many tens of times larger than microvilli. Cilia have active proteins in their core that allow them to wave, and this allows you to move things across the surface or allows the cell to swim along using these cilia in the external environment. So these are projections from cellular membrane. And the next organelle, as we call them, the next component we find in a cell is the nucleus. And most of us have heard of a nucleus. A nucleus is where the DNA resides in a cell. And the DNA is a single long strand. So you can sort of think of a nucleus more like the rubbery stuff in the middle of a golf ball. It's one long strand that's wrapped tightly around each other. And the image in the left and the right are just different views of nuclei. I'll just talk of the one to the left. In the pink one there, the little black dots, or the relatively middle-sized black dots you can see in the middle of each of the cells is the nucleus. Often the nucleus is found towards the middle of a cell. That's the common location you would see it. And the nucleus itself is surrounded in similar sorts of membrane, and that nucleus has the DNA in it, and in a while we will talk about the role of DNA and what it does. But it also, in that cell membrane around the nucleus, has little holes in it, or pores, that allows the materials to go from the nucleus to elsewhere. The next organelle I want to talk about is mitochondria. Mitochondria live inside the cell, and they convert inputs such as oxygen and nutrients into energy. And in a cell, energy is transmitted as ATP. ATP is a molecule that has chemical bonds in it that can release energy to allow other chemical reactions to happen. So a mitochondria is where ATP is produced, or it's reinvigorated from its ADP precursor. Mitochondria have a very interesting history. You might like to go and read about where your mitochondria came from as a separate exercise, but it is a really neat and interesting story. And the next organelle I want to talk about is called a ribosome. And ribosomes live on little strings called rough endoplasmic reticulum adjacent to the nucleus. And these ribosomes are responsible for making protein. And a ribosome makes protein from a template. And that template is called mRNA, and the template is produced as a duplicate of a particular piece of the DNA. That's called a gene, and we've all heard of what a gene is. Humans have about 20,000 genes, and each of them can be copied to a template called mRNA. That mRNA can then migrate out of the nucleus to the ribosome, sitting on the rough endoplasmic reticulum adjacent to the nucleus. And that ribosome can then read the template and produce the appropriate protein for that template. So messenger RNA has a very apt name, being messenger. It is really the message from the DNA, the controlling nucleus, to the ribosome to produce a particular protein. And what proteins are produced in a cell depends on their function. And up in the top left-hand corner, you can see a transmission electron microscope of millions and millions of ribosomes stuck along the surface of the rough endoplasmic reticulum. And then in the lower right, you can see a diagrammatic representation of those ribosomes, each one of them reading off the template messenger RNA, and then in the lower part, producing the appropriate protein from that messenger RNA. And once that protein is produced, one thing that can happen is that it can be wrapped up in a granule. And those granules, or vesicles being another name for them, those vesicles are really just little packets that can be moved around a cell using that cellular skeleton. Some of them may come to the surface cell and then squeeze out whatever protein was put inside the packet. So vesicles or granules are really canisters of proteins that have been produced by the ribosomes on the rough endoplasmic reticulum from the template messenger RNA, and that template messenger RNA was produced in the nucleus as a duplicate of a particular gene. And remember also that there are different mechanisms of moving across that cell membrane. As physicists, you would be aware of diffusion, the normal process of moving a chemical along its gradient from high concentration to low concentration. Remember the old trick of dropping a drop of coloured dye into a bath and watching it spread out. But there are occasions when you want a chemical to move against its gradient, and cell membrane has in its surface proteins that can actively transport against the gradient. Now clearly, to do that requires the usage of energy, and remember that energy is constructed in the mitochondria from oxygen and nutrients, and it's transmitted to the protein on the surface as ATP, and that ATP, by releasing its bonds, can allow chemicals to be transported against the natural diffusion gradient. So that's where I'm going to finish today. Other than, please, I've done a lot more videos on YouTube at Marc's Anatomy channel. Go and have a look and watch, and you'll see a whole bunch more about how cells produce proteins and what proteins can do. Thank you.